Hello, everyone. Welcome to our next edition of ICRN Translational Seminar Series. We are honored today to have Dr. Sergio Gradilone from Hormel Institute present his really exciting work on the role of cilia as a target in cancer in general and how it may relate to cholangiocarcinoma. Dr. Gradilone trained in Argentina at the National University of Rosario. This was followed by further postdoctoral work at the Mayo Clinic. He is currently an associate professor at the Hormel Institute. His work has been funded by the American Liver Foundation, PKD Foundation, the Fraternal Order of Eagles, and the National Institutes of Health. He's a member of many honorific societies, including American Society for Cell Biology, American Gastroenterological Association, and Association for Study of Liver Diseases. We are indeed delighted to have Dr. Gradilone present at this forum, and I'm sure everyone will find his work quite novel and exciting, and I have never seen an organelle being pursued as a therapeutic target, but I look forward to hearing his talk today. So thank you, Dr. Gradilone, for joining us.
Thank you so much, Mitej. I'm super happy to be here. When I contact you after the last Cholangiocarcinoma Foundation meeting, trying to seek possibility to interact more with this group, that was the idea, and you suggest to present here. So as you described, I'm in a research institute, the Hormel Institute, that belongs to the University of Minnesota. We are a standing alone institute that is off campus. We are like in Austin, Minnesota, 90 minutes drive from Minneapolis and 30 minutes drive from Rochester, Minnesota. So it's a nice research triangle that we have here in Southeast Minnesota. So my lab is a cell biology lab, and the idea of presenting here to you today is trying to find some new connections in order to be able to move forward our research more close to the patient. So as Mitej said, the title of the talk is Ciliotherapy, the primary cilium as a therapeutic target in carcinogenesis. For this audience, I don't need to spend time talking about the liver. So you all know the liver has two mainly epithelial cells, the hepatocytes that conform like 90% of the mass of the liver and produce the canalicular bile. And then the bile duct cells, the cholangiocytes, that conform the biliary tree. And these cells has these particular structures that are represented here as these small rods going up into the lumen. Those are the primary cilia. We can isolate and grow in vitro the cholangiocytes, and we can stain for ciliary markers. In this case, we stain with the ciliary marker is called acetylated alpha tubulin. It's one of the main components of the ciliary axon, and you can see that primary cilia is sticking out of the cells like an antenna. Cilia and flagella are really ancient cell structures. They have been known forever. You may be more familiar with the motile cilia. These, for example, you find them in the respiratory tract, in the reproductive tract, and they are hundreds of cilia per cell that move, and the main function is to move the fluids that are in the surface of the cells. Then you will have motile cilia in the sperm cells or in the unicellular algae chlamydomonas. And the other type of cilia that we know is the primary cilia or non-motile cilia. These are only one per cell, and they are expressed in almost every cells of our bodies. The main difference between these two types of cilia is the ultrastructure. So the motile cilia has what is known as a 9+2 ultrastructure. That means 9 pair of microtubules and a central pair conforming the ciliary axoneme, and that gives the mechanical connections to generate the force to move the cilia. Primary cilia or non-motile cilia is known as 9+0. That means that it's lacking the central pair, and that is why it's non-motile. So of course, cholangiocytes, as I mentioned, express primary cilia. This is an immunofluorescence of the cross-section of a bile duct of the rat liver, and you can see the cholangiocyte nuclei staining in blue, and we were super lucky here to catch a very long cilia in the lumen of this bile duct. And as I mentioned, if we look inside, we see 9 pair of microtubule structure. Keep in mind that in mammals, these microtubules are very stable because they are acetylated. So they are conformed by acetylated tubule. The presence of cilia in cholangiocytes was first described in the early 60s, and I always like to show this first abstract where the conclusion was that they may represent embryological remnants without a function. And that was actually the thinking at that time. Cilia was thought as an evolutionary junk without a function. Then in the 80s, Nick LaRusso lab at Mayo published some pictures showing the cilia in the cholangiocytes, but again, without evidence of any function. It wasn't until 2006 that LaRusso's lab got the first functional evidence of these organelles. This is an electron micrograph of rat liver, where you can see the portal vein, the hepatic artery, and a bile duct. And if we zoom in there in the bile duct, you can see all those hair-like structure in the lumen. Those are the primary cilia. So you can imagine bile flowing in this duct. Therefore, primary cilia are exposed to dynamic changes in bile flow, composition, tonicity, and they transmit those signals inside the cells. Right now, it's very well accepted that primary cilia is the cellular antenna. Even they made it to the New York Times. So it is now very well accepted that they function as multisensory organelles. During my postdoctoral work at Mayo Clinic in Nick LaRusso's lab, we learned that these organelles can function as mechanosensor, chemosensor, detecting different type of molecules like bile acids or nucleotides. Even they can detect vesicles. They can also sense the osmolarity of the fluid that is in the medium. So when at that time I was looking for my niche of research to start my independent path, we posed the question, what is the fate of primary cilia in tumors? What's going on with cilia in tumor biology? And of course, we went and assessed the expression of cilia in cholangiocarcinoma. And what we found is that primary cilia are usually lost in the tumor cells. These are immunofluorescence of a human normal liver, normal bile duct that is staining with CK19, so that's the green, and primary cilia in red in the lumen of these bile ducts. In the cholangiocarcinoma samples, this is a tumor sample, there is no cilia. So, and interestingly, we were able to reproduce this in vitro. So we can culture the cells in conditions that will favor the expression of primary cilia. And normal control cells, normal A69, for example, or NAC that is derived from a primary culture of cholangiocytes, compared to cholangiocarcinoma cell lines, like in this case, HUCT1 and KMCH, they do not express cilia. So we started to think what's going on, what happened when a cell is not able to express primary cilia. So we took a very simple approach. We took normal cells and removed the primary cilia by different approach. So a chemical approach, chemical deciliation using a drug, chloral hydrate, or molecular deciliation using knockdown of different genes that are important for the process of ciliogenesis. And then we assessed the phenotype of the cells. What we found at that time is that the experimental removal of the cilia, the deciliation of the cell, induce proliferation, induce migration. This is a scratch assay in real time, taken with the IncuCyte, and you can see that the normal cell that has cilia and the same normal cell that do not have cilia have increased migration. We also found increase in anchorage-independent growth, invasion assays, and the activation of signaling pathways that are normally correlate with tumor progression, like MAP kinases or Hedgehog signaling pathway, the downregulation of P53. So we started to propose the primary cilia as a tumor suppressor organelle. And I know it's a strange concept, but we are proposing that the multisensory functions of primary cilia are normally maintaining the expression of tumor suppressor signaling pathway and repressing the expression of tumor promoter signaling pathways. Therefore, we are proposing these ciliotherapies. So a therapeutic approach targeting the primary cilia. And we have two options for that. We can rescue the expression of primary cilia, or we can mimic the function of these organelles. For this, we need to learn, to rescue the expression, we need to learn the mechanism of ciliary loss. And to mimic the function, we need to understand the consequence of ciliary loss and understand what are the signaling pathways that are regulated by primary cilia. And then if we are lucky, we can move to animal models and hopefully sometime in the future to clinical trials. So regarding the mechanisms of ciliary loss, after screenings, we focus on HDAC6, histone deacetylase 6. HDAC6 is a unique member of the histone deacetylase family that is localized mainly in the cytoplasm and regulates the acetylation of non-histone targets, including alpha-tubulin. And as I mentioned before, alpha-tubulin is the main component of the ciliary axoneme that is acetylated to be stable. It was described that HDAC6 can promote the disassembly of primary cilia. Basically, what's going on is inducing the destabilization of the ciliary axoneme and the resorption of cilia. So we assessed the expression of cilia in tumor samples, and we found that HDAC6 is overexpressed in cholangiocarcinoma. So we started to think, okay, maybe this overexpression is responsible for the loss of primary cilia in the tumor cells. So we did a lot of biochemistry and cell biology experiments. Here I show you the overexpression of HDAC6 in a normal cell. So by Western blot, we show that we are overexpressing the protein, and that correlates with a decrease in the levels of acetylated alpha-tubulin, which correlate with a decrease in the expression of primary cilia in the normal cells. And that correlates with an increase in cell proliferation and increase in anchorage-independent growth. on the other side of the coin, we took tumor cells, cholangiocarcinoma cell lines, and downregulated HDAC6 using shRNAs. I'm showing here one of them that can reduce the expression of HDAC6 and increase the level of acetylated alpha-tubulin. And now the tumor cells start to express primary cilia. And that correlates with a decrease in cell proliferation and with a decrease in signaling pathways that I described before involved in tumorigenesis, like MAP kinases or Hedgehog signaling pathway. Then we move to a pharmacological approach. We try here tuvastatin A, that was at that time a very specific, fairly specific HDAC6 inhibitor at lower doses. So we treat the cells with this compound tuvastatin A, and after treatment, we start to see the expression of primary cilia in the surface of the cells. And that again correlated with a decrease in proliferation. But very important, I mean, that was key for us. This effect of tuvastatin A on proliferation depend on the ability of the cell to express primary cilia. So how we show that? We took these tumor cells and generating a stable transfection with one of the genes important for shRNA, for ciliogenesis. So these cells now are not able to grow cilia. When we treat the cells with tuvastatin A, even though we are inhibiting HDAC6 activity, we don't see much effect on proliferation, suggesting that the regrowth of primary cilia is important for the anti-proliferative effects. And a similar situation we found on the anchorage-independent assays. So we are proposing that HDAC6 is like that big wrecking ball that is destroying the ciliary antenna, the cellular antenna, and that helps the cell to for the malignant transformation, for the progression of the tumor. So we keep moving on the mechanisms of ciliary loss. So we had two different projects here. One was to understand why HDAC6 is overexpressed in tumor cells, and the other was to further understand the mechanisms by which HDAC6 removes cilia, and we end up describing this process of ciliophagy. So why is HDAC6 overexpressed in CCA? I will not go in much detail, but I show you the general idea. We found that this is a microRNA regulated overexpression. So what we found is that at least two microRNAs that target HDAC6, MIR433 and MIR22, are downregulated in the tumor cells. And we end up finding that these microRNAs, actually the precursor forms, I don't know if everyone is familiar with microRNAs. MicroRNAs are a small RNA of around 21 nucleotides that specifically bind to the target messenger RNA and induce the degradation of the messenger RNA or just inhibit the translation. So it's a mechanism to regulate the expression of different genes. So what we found is that, and these microRNAs are generated in the nucleus as a precursor form and then are transported to the cytoplasm where they are further processed to the mature form. What we found is that in tumor cells, the precursor forms were being trapped in the nucleus because of the effect on exportin 5. So basically the idea was that normally these microRNAs are being synthesized as precursors in the nucleus, transported to the cytoplasm process, and they go and inhibit HDAC6 expression. In the tumor cells, we found a defect on the expression of exportin 5. So these precursors were trapped in the nucleus and were not able to reach the cytoplasm and be generating the mature form to inhibit HDAC6. That's why HDAC6 is up and end up inducing the loss of primary cilia. So we were able to show that we can rescue this phenotype by overexpressing the mature form of these two microRNAs, or even by overexpressing exportin 5 and inducing that these precursor forms move from the nucleus to the cytoplasm. So this was a nice work done by a talented postdoc in the lab, Adrian Mancini. Then, as I mentioned, we are furthering our knowledge about the mechanism of HDAC6 dependent removal of cilia, and Stanislao, another postdoc in the lab, found this a ciliophagy process. So the increased autophagy in cholangiocarcinoma has been described in some reports in the literature, but anyways, at Nick Larusso lab, they helped us and they did the first screening in human samples, analyzing by electron microscope, analyzing the amount of autophagosomes in cholangiocarcinoma compared to normal bile duct, showing an increase in autophagosomes, suggesting that autophagy may be increased. Then we have a group of collaborators in Italy in the TGEM, and they analyzed the TCGA database, looking at a core of around 50 genes that are involved in autophagy, and they found that at least 35 of them are upregulated in the cholangiocarcinoma samples. And we also have data showing that in an animal model of cholangiocarcinoma, the rat orthotopic model of cholangiocarcinoma, the tumor had increased LC3 vesicles compared to the normal bile duct, suggesting that autophagy may be increased. We then further demonstrate that in vitro using comparing normal and cholangiocarcinoma cell lines, doing what is known as autophagy flags experiments, where you inhibit the lysosomal degradation of the proteins that are being targeted through the autophagy machinery by the use of buffelomycin or chloroquine, and you assess how the levels of LC3 change. And when you see increase of the levels of LC3, that means that there is an increase in the process of autophagy. And even more interesting, when we transfected normal and cholangiocarcinoma cells with an LC3 GFP, this LC3 is a protein that binds to the autophagosome when they are forming. So you can see the vesicles, the LC3 positive vesicles relating to changes in autophagy. And what was interesting to us was when we co-staining with ciliary proteins, in this case we use IFT88, we found that the autophagosome vesicles co-localize in the basal body of the primary cilia in normal cells, but even more interesting, in the tumor cells that do not have cilia, we can see the ciliary particles co-localizing with the autophagosome particles. Our collaborators in TGEM and Baylor College, Andrea Valavi and Nuncia, they developed a TFEB mice. These mice overexpress this transcription factor TFEB, that is a master regulator of autophagy that will induce autophagy, and they generated mice with the albumin cree. So there is increased autophagy in the livers, and interesting enough, these mice develop cholangiocarcinoma like tumors. And when we analyzed those tumors, we found that primary cilia are also decreased, suggesting that there is a relation between autophagy and ciliary loss. So we are proposing this ciliophagy mechanism where HDAC6 is removing the acetyl group from ciliary proteins and leave that lysine open to be ubiquitinated, and that protein is now recognized by some autophagy cargo receptors that will bind to LC3 and bring the ciliary components to the autophagy machinery. So we can then propose that by inhibiting HDAC6 and autophagy, we may have a good therapeutic approach. This experiment is showing tumor cells treated with an HDAC6 inhibitor or with an autophagy inhibitor. And in both cases, we see that we can increase the expression of primary cilia. And that expression of primary cilia induced by HDAC6 inhibition or by chloroquine, by autophagy inhibition or by the combination correlates with a decrease in cell proliferation. And we test this in different cell lines to be sure that that was a process that was happening in different cell lines. And in fact, we got similar data in the wheat cells, EGI. And again, we test if this effect of autophagy inhibition in proliferation was really important that primary cilia was present. So we did the same experiments. We have tumor cells that are not able to express primary cilia, and when we treat them with chloroquine in these experiments, we see decrease in proliferation, but not at the same extent that when the cell is able to regulate primary cilia. That means that at least partially the effects on proliferation of chloroquine are due to the ability to induce the expression of primary cilia. As I mentioned before, we wanted to identify what is the autophagy cargo receptor that would be responsible in this process of ciliophagy. For example, for mitophagy, it's described that the one that is important is P62. So Stanislao did a screening of the 4 more common autophagy cargo receptors, and he found that MBR1 is the one that most likely is participating in this process. So when he take normal cells and do a downregulation of MBR1, he can see increase in ciliary frequency, increase in ciliary length. And the same for cancer cells, HUCCT1 in this case. Downregulation of the expression of MBR1 induce, this is the picture, induce the expression of primary cilia. And when combined with an inhibition of HDAC6, in this case ACY1215, an HDAC6 inhibitor, the increase in frequency is even higher. So we partner with the Center for Drug Design in the main campus at the university. Bob Beans and Likian are helping us with this. We are developing a dual inhibitor. So we have generated an inhibitor that is designed to target both autophagy and HDAC6 at the same time. And we tested this in vitro, and we in fact found that the inhibitor, we are calling this Ciliomax, is able to affect autophagy and also increase the levels of acetylated alpha tubulin, that is a readout for inhibition of HDAC6 activity. This compound is able to increase the frequency and the length of primary cilia, and that also correlates with a decrease in cell proliferation and in colony formation. So going to the other side of the table, trying to mimic the function of primary cilia. We need to understand what are the signaling that are coming from the primary cilia. And there are a ton of signaling pathways that can be regulated by primary cilia. So the multisensory function of cilia depends on the enrichment, on the expression of specific transporters, channels that are expressed in the cilia. So Adrian, again, did a screening on the different signals that we usually work in the lab, and he found that nucleotides, in this case ATP, so a chemosensory function of primary cilia, is able to regulate the migration of the cells in a ciliary dependent manner. So he found that ATP in a normal cell that has cilia can decrease the migration of the cells. But in cells that are experimentally deciliated or in tumor cells that do not have cilia, the effect of ATP is the opposite. So this is a scratch assay to see migration. He tests the same to confirm using the InQSite with live imaging, and he see the same. ATP in the normal cells that do have cilia decrease migration, but do the opposite in the cells that do not have primary cilia. And a similar situation in invasion assays. He found that ATP can decrease the invasion of these cells that do have primary cilia, but has no effect or opposite effects in the cells that do not have cilia. So we can say that ATP regulates migration and invasion in a ciliary dependent manner. So he studied the mechanism of that, and he found that the main player is AKB1. AKB1 is a serine threonine kinase. Also, the correct name is STK11, and is described as a tumor suppressor involved in cell polarity, migration, invasion. So he did knockdown of this protein, AKB1, and he found that when AKB1 was downregulated, the effect of ATP on migration was abolished. So that we start to think, okay, yeah, this is the pathway. He also assessed if this protein was phosphorylated, because the active form of AKB1 is phosphorylated in serine 428. So he found that the activation of AKB1 induced by ATP was only happening in the cells that do have primary cilia. When there is no cilia in the cells, we don't see that activation. Moving forward, he assessed if this effect is dependent on purinergic receptors and on pKa. So in fact, doing biochemistry, he demonstrated that. Here I show you a Western blot where you can see that ATP activate AKB1, but when he used suramine, that is a general inhibitor of purinergic receptor, or H89, a pKa inhibitor, we don't see the activation of AKB1 induced by ATP. So then he needed to identify the purinergic receptor responsible for this signal, and based on the pKa response and the human purinergic receptor expressing cholangiocyte, there were at least two possibilities, P2Y1 and P2Y11. So he did knockdown of these two genes and found that P2Y1 has no effect on the phosphorylation of AKB1 induced by ATP, still happening, but the downregulation of P2Y11 actually abolish the AKB1 phosphorylation induced by ATP, suggesting that P2Y11 is the one. And also on the functional side, he showed that the P2Y11 downregulated cells do not respond to the ATP treatment. And he ended up showing that P2Y11 is expressed in the primary cilia. So this is a little bit the working model of this project. Normally, primary cilia is detecting nucleotides by P2Y11, and this by a mechanism that involves an adenine late cyclase 5 and pKa, end up activating AKB1, which in turn activate P10, and this end up inhibiting AKT and the activation of the polymerization of F-actin and inhibiting the migration of the cells. So when there is upon malignant transformation, there is loss of primary cilia. So we are losing this activation of AKB1 by nucleotides. Therefore, AKT is now active and able to promote migration. So his next step was to look for an activation of AKB1 in a way to bypass the need of primary cilia. So he found this natural derived compound HMC, is hesperidine methylchalcone, that is able to activate AKB1 independently of the expression of primary cilia. So you can see here that can activate AKB1 in cells that do not have cilia, and also is able to reduce the proliferation and migration in the tumor cells. And of course, this is a specific, fairly specific process, because when he knocked down AKB1, the effect of hesperidine methylchalcone in migration is not there. So basically, he's showing that by activating AKB1, he's able to bypass the need of primary cilia to activate AKB1 and is able to reduce the migration of the cells. So when we move to the preclinical animal models, we have been using in the lab for the last few years a rat orthotopic model developed by Al Sirica. He kindly provide the cells, the BD cells, and we are able to test our ideas on this model. So here I'm showing you the treatment with tuvastatin A, that is one of the HDAC6 inhibitors, showing that when we treat the animals with tuvastatin A, we reduce the growth of the tumors, as you can see in the bar graph. And interestingly enough, the tumors the control tumors do not have cilia, but the smaller tumors in the tuvastatin treated animals, we can find primary cilia expression. In this experiment, we treated the animals with a different HDAC6 inhibitor, in this case ACY1215, that is even more specific. And also we use the autophagy inhibitor hydroxychloroquine, or the combination, and we found that indeed we can reduce the tumor growth with these treatments. And finally, as I just show you, hesperidine methylchalcone, HMC, is able to reduce the growth of the tumors in this model. So what I'm showing you today is that to move forward these ciliotherapies, we need to identify targets to rescue the expression of primary cilia or to mimic the function of primary cilia. So to rescue the expression, we have identified that HDAC6 inhibition is a potential approach. There are several inhibitors that are in clinical trials now for other diseases, so it would be important to test. Autophagy inhibition is another approach, and we are now pursuing the combination of HDAC6 inhibition and autophagy inhibition with this compound Ciliomax. We have also identified microRNAs that we can overexpress in the tumor cell with the use of AAVs, for example, or we can overexpress exportin 5 to rescue the transport of the microRNAs to the cytoplasm and get them functional. On the other side, trying to mimic the function, we have found that the activation of AKB1 could be a potential road to go with the HMC. And we are currently, let's talk about this general working model. So basically what we are trying to show, and what is our hypothesis here, is that normally the multisensory function of primary cilia works by activating tumor suppressor signaling pathway like p53, AKB1, and by inhibiting tumor promoter signaling pathways like MAP kinesis or Hedgehog signaling pathway. So we can call primary cilia a tumor suppressor organelle. But upon malignant transformation, by a mechanism that we are starting to understand that involves downregulation of exportin 5, downregulation of MIR22 and 433, which in turn induce the overexpression of HDAC6, and then the loss of primary cilia that we now know that also involve the process of autophagy. So when there is loss of primary cilia, there is a disengagement between the environment and the cell interior. Therefore, we are seeing downregulation of p53, of LKB1 that are tumor suppressors, and we are seeing overexpression of or activation of MAP kinesis and Hedgehog signaling pathway. Therefore, if we can restore the expression of primary cilia, for example, by targeting HDAC6, or if we can mimic the function, for example, by activating AKB1, we may turn the cells to a more normal-like phenotype and reduce the growth of the tumors or even induce cell death. And we are super excited about this because the loss of primary cilia now is not only described in cholangiocarcinoma, but in other tumors like pancreas, breast cancer, melanoma, prostate, colon cancer, which make our basic research more broadly applicable to other diseases. And as future directions, we keep working, trying to understand the mechanisms of ciliary loss, and something that we are working now, getting ready to submit the manuscript. We found that SIRT1 is another deacetylase, is also involved in the loss of primary cilia, and we can inhibit this using the drug sirtinol. And on the consequence of ciliary loss, we have a few projects, very interesting projects right now. One that is studying the role of primary cilia regulating the glucose metabolism of the cells. And then the second one that is, it's in an embryonic stage now, but it's super exciting, is we are studying the connection between the primary cilia with the nucleus and how primary cilia can be regulating epigenetics and DNA repair. So just some thanks here, of course, to funding and Nick LaRusso's lab and Louis Robert. Louis has shared with us tissue microarrays that we are not using. Dr. Kabir Modi, that he was excellent, very helpful. He prepared actually a clinical trial for an HDAC6 inhibitor, ACY1215. Everything was ready to go. Unfortunately, the company that has the compound was sold, and there were some financial problems, and we couldn't move forward. Then Chus Bañales in Spain that provides cell lines, and they have a nice database in the ENSCA that we have access to, and our collaborators in Italy. Thank you so much, and happy to answer any questions.
Okay, well, a virtual round of applause for Sergio. Very, very fascinating indeed. There were a few questions here, and I want to encourage anyone with questions to please just put them in the chat box so we can query Dr. Gradilone while he's with us. Can you speak to the heterogeneity of expression of or loss of cilia in cholangiocarcinoma cell lines and other models and patient samples?
Yeah, that's a fantastic question. So that's something that we need to explore a lot more. Regarding the human samples, we have our first study that was very small, with around 30 patients, and we found that the loss of primary cilia was around in 70% of the patients. But of course, we need um, a bigger study with that. That's why I'm here and trying to get connections, because we need a bigger study showing, assessing the expression of primary cilia in tumor samples, and see if we can correlate that with different type of tumors and with prognosis on the patients. On cell lines, the the few that we have in the lab has no cilia mostly. I mean, the expression is low. Sometimes you can get up to 5% of the cells with cilia when you compare with a normal cell that you can go up to 50, even 70%. Those are HUCT1, KMCH, EGA, WHEAT, but there are other cells that you will find cilia. So we have some that we are studying and see if there is any correlation with the different mechanisms, expression of HDAC6, expression of SIRT1, why we see that. But of course, there is will be a lot of heterogeneity there that we need to study and understand.
Okay, thank you. There's another question pertaining to relationship between HDAC6 and Aurora A kinase and PLK1. How are they interrelated in ciliary expression and carcinogenesis?
Yeah, Aurora A actually is a kinase that would activate HDAC6, so they are closely related. Uh, so definitely there is a relationship there. Yeah.
Okay. Question from Dr. Benales actually. How can this potentially be applied to SARS-CoV-2 infection, given the role of ciliary regulation in I guess entry of the virus into the cells?
Yeah, no, that's a great question, Chus. Yeah, we actually put an internal proposal trying to start doing some experiment on this, studying the expression of the AC2, yeah, the receptor in the primary cilia, and what happens when the primary cilia is lost. Yeah, but unfortunately, we haven't advanced on that yet. The idea is there, is worth to explore, definitely.
Got it. Can you use the work you described in some capacity to um, better inform diagnostics or as a prognostic marker?
We need to study that. Definitely, that would be the goal for a collaboration with a clinical lab that have access to samples. We would like to see if we really kind of identify any correlation between the expression of cilia and the prognosis, or the levels of HDAC6 and the prognosis. Yeah, definitely.
Got it. I had a general question along the lines of what you presented. So you have cilia and then loss of cilia, and with cancers, one of the mechanisms people talk about is loss of contact inhibition. Do you feel the cilia play a role in that? And when you lose that, that's actually a surrogate for loss of contact inhibition?
Yeah, that's definitely. So primary cilia could be regulating cell-cell interaction and would regulate cell polarity. So the loss of cilia will contribute to that. Yeah.
Mm-hmm. And further along those lines, at the cell level, you have the contact inhibition, and then at the organ level, you have things like the hippo-YAP pathway. So do these pathways interact somehow in terms of, again, the hippo-YAP pathway, or not really?
Uh, I don't have an answer yet to that. We are starting to explore. We got some samples from Dr. Risby and Gors at Mayo. They have a model with a YAP activation-derived tumors. So we are studying the expression of primary cilia there, and we looking for interactions with our pathways. Yeah. Hopefully, I can answer that sooner.
No, it's more a sort of very hypothetical question at this point, I suppose. Maybe one last question here, and then we can wrap up. Can you comment on sort of translation to the clinic for chloroquine from the perspective that you described?
Yeah, definitely I see potential there. So in the animal, in vitro and in the animal study, chloroquine alone is giving us reductions in tumor growth. We are actually developing this dual inhibitor, honestly, basically to have a new compound that we can get funds to support a clinical trial.
Yeah, because you could sort of independently inhibit autophagy and HDAC6. You're
Yeah,
just sort of
yeah.
putting it into one compound.
Yeah. I mean, I have not much experience with clinical trials, but from what I'm hearing from the chemists at the main campus and other collaborators is that actually you need a compound with a patent to get actually the trials going. So it looks like nobody will fund a trial on a drug that there is no potential business there,
Right.
unfortunately.
Yeah. Most of the funding does come from biopharma and the private sector.
Exactly.
So you're
Exactly.
right. Okay. Well, again, it has been a very exciting hour with you. We can clearly see that the story is still evolving, and we hope to interface with you more in ICRN and maybe help advance some of these ideas to the clinic and learn more and hopefully benefit patients as we go along. So thank you so much for joining us today, and
Thank you for the
I've
opportunity.
learned a lot myself here.
Thank you so much.
Okay, take care. Have a great day.
Bye bye.
Bye bye.